Okay? So what I'd like to do today is continue on. Remember we started talking about seed banks and seed pools and you know, how long do these things survive and what are their numbers? And I'm just continuing on here. Again, there's no new handout. I'm continuing on chapter 3. Okay? And take a look at some of those numbers for arable land in terms of the number of seeds per meter square. Okay? You all know a meter is about 3.3 feet, so one way. So look at the numbers that typically you find in arable land. Okay? Tropical crop fields, 7,600. It's not, okay? And these are just ranges, but it gives you a sense that by far arable lands are have the highest quantities of, in this case, weed seeds or seeds. Can anybody tell me why? Does that surprise you or it does not surprise you relative to some of these other systems? Usually when we're talking seed banks, mostly it'll be about a 6-inch depth, 15 centimeters. That's the standard when we do it. I mean, by far, you know, 80% of them are going to be within the first inch to 2 inches of soil. Okay? So up to 10 centimeters. But the standard sampling protocol brings you down to 6 inches, if you can. I mean, some of the areas in the Alvars south of Lake Ontario, we can't even get 2 inches into the ground and we hit, you know, limestone. So, but if you're in an arable field, that's typically the case. So does that surprise you? Maybe in terms of not necessarily, you know, it's 34,000, but just relative to these other systems. Why or does it surprise you and why does it not surprise you that it's that high? Kyle?
Banks don't have winter, so seeds get dispersed and it's grown now, or they don't have a lot of dormancy. It's
right.
Long-lived seeds.
So I mean, there's just much more of a recycling. I mean, there's a lot of seeds that'll go in, but more, you know, a lot will be coming out. They're not going to be sitting there at any one time. You sample and they're, you know, whereas this is an accumulation in temperate areas, and I should say, you know, temperate arable areas. But what else is typical of arable land? What have we talked about? See, they're the most fertile.
Lots of
Disturbance, and what habit type species are adapted to this?
Mostly
Annuals, right? I mean, that's why. So it's annuals. What are they known for? There are strategists, competitors, rurals. They can get in there. They emphasize resource allocation to productivity, to reproduction. Okay? They're not there for the long term. They are, in a sense, by passing on seeds to the next generation, but they themselves physically are there 3 months and they're gone. So if you go out in the fields now, look at all that stuff going to seed. Okay? So again, I wouldn't be asking you, tell me, you know, exactly what that is, what the numbers are, but just as a relative, understand that we're dealing with a large number, high quantities of seed, particularly if, you know, these things can survive in soil for a long period of time. Okay? We break it down into some of the, and again, it's millions of seeds per hectare. Okay? How many acres is a hectare? 2.48, that range, just to give you an idea. Okay? Again, not looking at the numbers, but just, I mean, I wish we had this even in, you know, just millions per acre because the number just explodes. But just the numbers are astounding. Look at that annual bluegrass we talked about in class, you know, 16.6 million seeds per hectare. I mean, and again, they're usually going down about 15 centimeters, okay? So really problematic. Some of these species, you know, we don't typically have here. This is out of the UK. Okay, and it doesn't matter, it's 1968, it could be 1958, it hasn't changed any. A lot of them are still there. Okay? So, here is just impressing upon you the quantities. And the longer lived some of these seeds are, the more they're going to be accumulated in there. Okay? And we're going to talk about dormancy and what that means. So, and again, you see the standard 6-inch, 15 centimeter as being a standard for SAM. So if any of you have to do seed bank studies, think about that. Soil cores, you want to know how many seeds are in a volume of soil. And so you need to have the depth, diameter. Okay? So, how long do some of these things live in the soil? Okay? Again, don't get caught up in all the numbers, but just see the trends, what we're getting at. Let's look at velvet leaf, since it's one that most of us know. Okay? Mean percentage of original population of wheat seeds still viable after burial for up to 5.5 years. Which isn't that much, but it's still 5 years. Okay? Look at velvet leaf. When it starts out, they're 100% viable. After 1.5 years, okay, 87% of the original seed batch is still viable. Okay? Drops to 65% after 3.5 years. After 5.5 years, almost 6 years, you still have 36% of the seeds being viable. You say, well, there's still a whole lot that are dead or germinated. But think about that each plant can produce a really, fair-sized velvety plant can produce 40,000 seeds. That, one plant. So you can imagine, okay? Some don't have very much, in terms of, here's Pit and Morning Glory, the Epomia, okay, down to 13%. Typically speaking, grasses, typically, except here's Johnson grass at 48%. Generally speaking, grasses do not survive long in soil. That's one of the advantages. Annual grass seeds, just because of their structure, are more susceptible to decay and disease and burial, basically, if they go too deep. So that's one advantage. Whereas the hard-seeded species, the morning glories can do that. Okay. The same thing for pigweed. It's actually interesting to see that only 1% of seeds after 5 years are still viable. However, each pigweed plant on average can produce 250,000 seeds. 100,000 to 250,000 seeds. So you see what you're against. That's why we need to talk about this. So the take-home message here is that in general, high percentage, okay, of seeds can remain viable in soil. Particularly true of hard-seeded dicot species. Okay? However, as I mentioned in previous classes, don't get the idea that 90% of seeds will survive of a given species survive to 40 years. Because you hear that. You say, oh, velvety can survive 40 years. It's a small percentage, 0.1% or something. But again, keeping in mind the numbers, the productivity, number of seeds that are produced, this becomes pretty important. Yes?
The study seems odd with the different depths that they're looking at. How did the viability after 1.5 years, how would those numbers look if they hadn't buried the seeds so deep?
The general trend with burial is that the deeper you are, the longer the viability in general. Just because things can get to you. I mean, over time, that's one, it's an advantage and disadvantage when we're cultivating. So when we're moldboard plowing, and let's say we've had a bad year, we've been pretty good, and I'll come, this will tell you to your question, Bob, is we've got seeds one year, we just couldn't get in there, it was wet, or, the grower was sick, couldn't get in there, and we have a high quantities of seeds getting in there. Then we go in and we moldboard plow, which inverts the soil and basically takes the top layer and puts it down 1.5 feet or something. Now those seeds are most seeds of those seeds are too far down to germinate. So that's a good strategy if you've been good about managing weeds. Basically keep them down there. Don't bring them back up. However, the one disadvantage is being that far down, soil can be a better buffer for them in general. Now, the really tiny seeds, they're just too deep. Most seeds are going to be too deep to germinate. If you wouldn't disturb that soil, they'd be viable, but they wouldn't be coming up at all. You wouldn't be touching. But that's not what happens. We go the next year and we're moldboard plowing again. And what do? We take that bottom layer and bring that up, take the top layer and bring it down. Now, given that I just told you that these seeds, most of them are still going to remain viable, okay, then that's kind of a problem. It's out of sight for that 1 year, but they are going to remain more viable. So with increasing depth in general, viability will increase. Okay? Eventually they will, they're going to be starved. Okay? But what I would have liked to have seen is if they had broken this down. And I'll show you the viability relative to depth in some species, how that varies. Okay? So it's an advantage to bury things deep, to say, okay, I know they don't have enough resources in the seed to come on up. But if I go back the next year and bring that soil back up, don't think, oh, they stayed there for a year and they're dead. They're not dead. Most of them are going to be viable. Okay, so that's why in no-till we try to, or stale seed bed, we just scratch the surface, basically the first inch or two, to basically stimulate germination, wipe those out. But afterwards, when we plant, we don't start mixing that soil again, because we've now just at least sterilized, as I would say, to the best of our abilities, that 1 inch or 2 inch, okay, depth. where most of our wheat seeds are going to be. Okay? You don't want to go back in there and dig. And that's one of the main problems that gardeners, that I noticed in their gardens. They go in and they've got their shovels and they're turning the soil over and over again. They're bringing seeds. Often I tell them, just, you know, I'm not saying do no-till, but just sterilize the surface. Just basically rake it in, allow some of the seedlings to come out, and then don't do anything. Go in and plant as almost in no-till. The more disturbance, the more you're going to get problems with these weeds. Okay? So, here's some general characteristics of seed banks that I'd like you to be aware of. And most of this we've covered, but I just wanted to at least have it there for you. Okay? Pollinating species, which are weeds, are early successional species, will contribute more seeds to the seed bank than later successional species. No surprise here. These guys are producing 100,000 seeds per plant. You get down to the grasses and you then get shrubs, they're not producing 100,000 seeds. You know, birch is not going to do that, and even the poas. Okay, most of them are going to be vegetated. They're competitors. They're really there to compete, not to produce seeds. In natural systems, this is neat. If you actually go into a forest that hasn't been disturbed and you sample the soil, okay, and you try to germinate whatever's in there, in that seed bank, and you then compare it to what's above ground, often there isn't a very good correlation. But in ag systems, there's a very strong correlation. What's above is going to be telling you what's below, the iceberg, the tip. Okay? There's little correlation between the active and passive populations. Remember, the active is what you're seeing above ground. The passive is the below ground. In some cases, these are dormant. Okay? You reap what you sow, basically. And like I said, if I go to, I just saw my lawn again today, and it's purple because I've got crabgrass, the Digitaria, Sanguinalis initium, both species, they're just there. I mean, and every day I'm going about, these seeds are going back into the ground. I need to cut them off. Because if I don't do that next year, I know what that turf is going to look like. Okay? Questions? Okay. Now, this seed bank remains relatively constant. Okay? Especially if species are allowed to complete the life cycle. Okay? Now, we don't want that. We don't want it. We want to draw it down. That's part of what we're going to be trying to do. But if you allow the species to complete their life cycle, they're going to be in there. So what this is saying is, if I go in the field and I'm seeing velvet leaf pigweed, quackgrass, particularly mostly annuals, let's say barnyard grass, a lot of them, that's an indication I've got probably a lot of seeds of that species down below. And they're going to be with me for a while. So use that as a guide to tell you, if I've got one or two seedlings, then that's a good sign. But when I have this flush, something's happened there. Okay? Now, seed banks act as a buffer. Okay? They don't just destroy the seeds. Okay? The seed bank is slower to change than the active population. It acts as a buffer. Remember we talked about what's the advantage of having, susceptible species of, let's say we have lamb's quarters. We do have lots of lamb's quarter here in New York State that's resistant to atrazine, our major corn herbicide. Okay? And it's developed over time. But in our seed bank, we may have a whole bunch of seeds from 15, 20 years ago, okay, that were susceptible. They never were exposed to the atrazine, although that's not, most of it in New York sprays atrazine, and they've been doing so since the mid-60s. But what I'm saying is that the genetic pool in the seed bank, because the soil seed bank acts as a buffer, okay, will provide you with more genotypes, okay? Remember, each seed is an individual genotype. It's not a clonal. It's not part of a clonal species, okay? And what this does is that ensures population species survive even in unpredictable conditions. So if, for example, we have a really, we get snow in June, wipes all of these weeds out. Our crops, we have to go back and replant or decide we're not going to be planting. Just think about the floods in Iowa, in the Midwest this year. A lot of the growers never planted. Okay? It wiped out, it also choked off a lot of the weeds. They were up, and some of them couldn't, they just can't handle anaerobic conditions. That seed bank, because it's probably got a lot of seeds in there, even in those areas, it's going to be back next year. It lost the top, so it's a way to ensure survival for a species. So it's kind of like a little genetic bank. Okay? That's why these plants. Now remember, for cultivated plants, we bred out all the dormancy because we can't afford to plant 50. we're putting down 30,000, seeds of corn per acre thinking we're going to get 30,000 plants. That's the seeding rate we want, planting rate, and say only half germinate. That's not what you want. You want 95, 98. So that's been bred out of our cultivated species, but not out of the weeds. So you're never going to get 100% germination. In fact, sometimes you get 20%. Ragweed, common ragweed, you're lucky if you get 20% germination from any 1 year's production. And I'll talk to you about what it does. Why is it dormant? What's going on? Okay? Seed longevity in soil is longer than in storage. So if you take seeds and you put them in, vials, and we do that often. We're trying to grow during the winter, grow these things. We have them in paper bags. We put them in the fridge. Nothing's as good as soil. The best thing is you put them in soil. Okay? And so this is very important. The other thing is that when seeds are in the soil and I say they're dormant, they are not dead. Okay? In fact, they're very much alive, as I mentioned last class, and are very much in contact with the environment, always kind of getting a feel for what the temperature fluctuations are, what the humidity is within the soil. And there are basically going to be some cues that are going to allow some of these seeds to germinate, and we'll talk about what those are. Okay? So, here's some other important aspects, and this is really important for ag, for management, is that cultivation redistributes seeds throughout the seed profile, and I will show you what a seed profile looks like. If you're using no-till or minimum till, you're basically going to have the top part, okay, we're basically going to have your seeds, and then it's going to decrease. If you moldboard plow, you're going to have throughout your soil column a redistribution of the wheat seeds. They're going to be so. And this is true actually even for if you're trying to control something like quackgrass with rhizomes and distributing them throughout the fields. Okay? Now you could use cultivation to an advantage. As I mentioned, you could moldboard plow. If you've got a bad year, you got a lot of weed seeds, you say, I can't handle this, whether you use herbicides or not, say, I'm just going to bury them deep enough that they're not going to be a problem this coming year. Okay? And that cultivation can do that. Okay? As I said, the problem is if you go back to next year and re-moldboard plow, you're going to be bringing those guys back up now. And the idea is that they're not dead. Most of them are still alive. They would have to be under there for a few years, as you saw, maybe 2, 3 years, but so the idea would be maybe then to just cultivate in a shallow fashion. Don't go down, maybe chisel plow or something. Okay, and we'll talk about that. Okay, no-till systems do not redistribute seeds through the soil profile. You'll see the soil profile. Most of the seeds are going to be right at the top, and that's why we need herbicides, because they're going to be germinating, and we don't have any cultivation. How else are we going to control them? So you need to use herbicides. But the nice thing is that they're basically just on the soil surface, and you can take care of those. Okay? So, what is the cultivation action? We'll talk about this, just to give you an idea. What would these do to a seed bank? In general, in our annual crops, what would this do? And this is something that you could think about. So, it would be a nice prelim type question. I'll cover more mechanical control, but just in your own minds, and for those of you who may not be familiar, what is a chisel plow? I'll go over that. What's conservation tillage and so forth. But just understand that kind of moldboard plow is an implement that causes the greatest inversion of soil. It really disrupts the soil, and you get these major changes in the soil structure too. We'll talk about that. Okay, this is showing you chemical control, yearly tillage. Okay, what it's showing is velvet leaf seeds, number per hectare, and again, you may not have this figure. If you don't have it in your handouts, don't worry. I mean, I'm just trying to show you a trend here. Okay, so this is in millions of seeds and number of years, and they basically in chemical fallow and yearly tillage. So chemical fallow is you're basically not planting any crop. They just wanted to see how long would it take to draw down the seed bank. Okay, and they would go in and basically spray a herbicide to kill the velvet leaf. And clearly, over the number of years, okay, here's the round circles, okay, you will draw it down. And still it's interesting that after 16 years, you still haven't drawn it down to 0. Like I told you, there are going to be some seeds of velvet leaf that can't survive up to 35 years. Okay, and for those of you in the dairy, interested in dairy, the seeds go right through the gut of cattle. And when you spread manure on your fields, if they're not properly composted, a good percentage, maybe 20, 30% of the velvet leaf is still viable when it comes out of the animal. Chickens is another story. They really do a number on the, or if you have to compost over 150 degrees Fahrenheit. So you really have to get those temperatures high enough in the core. Okay? Otherwise, you're going to be spreading the problem. Okay? But again, this is showing that even chemical fallow, okay, you're not putting a crop, you're just each year you're making sure the seeds are no new seeds are coming in. This is the idea here. You're drawing down. How long will it take to draw down this population that starts off up here at 120 million per hectare? Okay? And you notice that actually yearly tillage does a pretty good job. Okay? Yes?
But yearly tillage for almost 18 years has a lot of negative.
Oh, this is strictly academic. You're absolutely right. From a practical perspective, nobody's going to do this. No, you're absolutely right. I mean, chopping it up, but it's just showing, and the reason why, so yeah, this is not, and we'll talk about what are some of the management, that are, and some of them will need to balance soil erosion issues, soil health, soil stability versus management, okay? So by no means did I mean that this is something, this was just to give you an idea of what are we battling here. Even when we don't allow any new weed seeds to go in, it's still taking us a fair amount of time, at least with this species under these conditions. Does anybody know what the reason might be as to why we draw down the seed bank much faster when we're with yearly cultivation? Does anybody have any idea? Rather than just, putting down the herbicide.
Disturbing the ground, more seeds will come each year.
Exactly. You're disturbing the ground. Remember, and we'll talk about what stimulates germination. One of them is aeration, bringing them up to the soil surface, exposing them to light. When seeds are close to the soil surface, what happens in terms of temperatures? Day-night temperatures. They're going to be closer to the surface. A lot of them detect the fluctuation. Cool nights, warm days. That's why in the spring they germinate, is we get those cool nights, kind of what we're getting now, basically. And that's why some of our winter annuals are germinating now. Cool nights. I mean, we were down to 40 last night in Dryden, where I live. But today we'll be up to 72, 74 degrees. That kind of, the bigger the alteration for many tempered weed seeds, that is definitely a stimulus for germination. And in many cases, light is also a stimulant. Not for all. We have some species that actually don't need light. They'll germinate under dark conditions. And we'll talk a bit about that. But, right, the key here is that you're basically disturbing the soil. Okay? Having just said that, this is just a theoretical, diagram. Okay? And what I wanted to impress upon you is how difficult, even if we get most seeds to germinate, that's in the seed bank, but we miss a few, it's still going to take us quite a bit to basically draw down the seed bank. Okay? And what this graph is showing you here is number of propagules, hypothetical, hectare per hectare, millions per hectare. And here's the number of years. And so this is calculated time needed to eliminate viable weed seeds from the soil under different levels of germination and control. Remember, I told you the ideal would be if we get 100% germination over a number of years and we kill all the weed seeds. That doesn't happen in nature, even in theoretical conditions. This would be our kind of best scenario. I'll start out here. We're 98%, so let's just say we could get pretty well all of the weed seeds in the seed bank, except for 2% of them, to germinate. And when they come up and emerge, we wipe them all out. We make sure, we would be sure that none of them would go to seed. If we start at these levels with this is, again, theoretical based on some modeling, in 1 year's time, 1 or 2 years, we could draw this population down. We'd have to make sure no new seeds are coming in from elsewhere. You're not bringing compost. You're not bringing mulch. That's bringing it. Let's say this was a perfect system. I've got my little flat there, and I know how many seeds in, and I get them for whatever. We never get 98% germination. Never. And I'll show you why, because each of these species has different mechanisms for dormancy. But it would be. Now, let's move here. Now we only get 75% germination. It's still way higher than we typically get in our fields. And, we basically kill pretty well all the propagules and control them. But, again, 25% of them are not going to germinate. They're going to be in the ground, and they're going to be proportionally be coming out later on. We're down to about 3 to 4 years. 75% germination, so it's very similar to this, but what happens is you don't kill the 100% that come up. And again, it'll vary by species, how many seeds are produced, but this is true of many of our annual weed seeds. Look, we missed 0.5% of the ones that came up. Okay? So 25% didn't germinate. They're still in the ground. Of those that came up, we just missed 0.5%. One. Let's say we missed one plant. Okay? It's now going to take us almost 10 years to draw it down. In the real world, this number here, it's probably no more than 20%. 20% of our seed bank would even germinate in a given year, even under our best conditions of tillage and so forth. And even in Roundup-ready systems that, it's pretty good control, we still have escapes. Whether they're resistant or whether, the spraying just didn't go, it's pretty good control, but we never get 100% kill. We'll still get seeds going in. So that is one reason why, you're going to be controlling weeds for a long time, unless you find a way to get all that. This needs to be 100% germination, 100% kill. It hasn't happened yet. In all 10,000 years we've been battling and beyond. Okay? Just to show you how important the seed bank is for managing weeds. Most weed scientists agree that more focus should be put towards understanding that seed bank, because that's where we can really, make a difference. Getting them in. I mean, the seed bank, and ideally would be, and what I mean there too is seed rain, not allowing things to go to seed in the first place, would be the ideal. And that's why I think I mentioned, did I mention to the group this new machine that John Deere is working on that's basically going to be sandblasting seeds in the combine? That would be the kind of thing, because that's going to, you can get high germination, but it's the numbers of seeds that are in those seed banks that are scary. And that there's, so it has to be something that basically is going to really put a dent in the number of seeds that are going in the ground, and then you doing a good job of controlling them. If it's missed here and there, you put down the herbicide, but a couple of rows got missed, or the herbicide wasn't effective, we didn't get rain, and we put a pre-emergent herbicide down, that happens. Or even in cultivation, it was too wet, we couldn't get in there, the weeds got away from us, they're too big, they've set seed, you're back to square one. I mean, that's the problem. It's not that the growers aren't trying, and but, okay? I won't say much about this, but I just wanted to show you that for weed seeds, this is showing you what a seed bank for arable lands would look like. And the dark, part of this is just showing, this is the months of the year. The dark part is showing you that if I would go in an arable field and sample, let's say I could do this throughout the year and sample, I would find a basically persistent seed bank. There will always be seeds there of weeds in temperate arable lands. This is not true of some of the other areas that, like the tropics, for example, that have peaks and bounds. I'm not saying you could go in there in arable land and not find any weed seeds, but what's the problem with our system, our arable system, is just the number of seeds that are going in there. So yes, things are going to germinate, and that's what this white area indicates, that a given time of year, for example, in our case, it would be in May, June. We get most of our summer annuals germinating. Yes, a lot of them would germinate, and so they peak. But you'd also have some that are produced, a lot that are produced that are dormant coming off the plants. So when seeds are produced and you pull them off, so if you go now and you grab the foxtail seeds, any of these seeds, and you just give them the perfect conditions for germination, give them light, give them water, give them temperature, a lot of them are not going to germinate. They are dormant. Okay? Does anybody know what the advantage? Why wouldn't they germinate? Sorry? Right. That's an advantage. Why? What are you going to germinate in September and October? It's getting late. Our winter annuals already have germinated in most cases. They can survive. But our summer annuals, it's basically a death trap. You're not going to make it, not under these conditions. Okay? So this is really the type 4 kind of seed bank is the one that I'd like you to focus. This is the persistent seed bank that's maintained by early colonizer. These other ones are, in terms of annual grasses, type 3 and 4 are really the big ones. Often these are combined together. But there is, what I'm trying to get at, it there's no period of the year where I go in and I sample and I say, oh, I'm not finding velvet leaf this year. Or at this time of year, it's either all germinated. No. Even in May, even in July, even now, I can go in and I will find velvet leaf seeds for you. So persistent. That's different from transient seed banks. Some species have transient seed banks, mostly tropical species. They're in the soil for a short period of time because they don't have dormancy. As soon as the right environmental conditions go, they produce seed. Pretty well all of them germinate. Okay? They produce seed. So if I'd go right after and sample, I'd find very few of them. Okay? And then, of course, they would produce more seeds, and of course, then there'd be a spike in the seed bank. But there are times of the year where you don't find large quantities. That is not true of our arable systems. Absolutely not true. Okay? That's what I wanted to impress upon you with this. Just, again, building up the problem. So now it's, you know, you're starting to see this is majorly problem. Of course, we'll talk about management strategy. What do? We can't just throw up our hands and say, ah, clearly we don't do that. Okay? So this is, okay. So let's get into that seed bank model. You know, let's start thinking, and we started that last class. Let's start thinking about what happens to these seeds in the seed bank. Okay? So this is going on right now. We've got seeds that are maturing on the plants. They're ready. Think about any of your weed species. We were out with the IPM group on Monday. What did we see? We saw all kinds of, in the alfalfa, you know, patch. We had pigweed. We had large, you know, crabgrass. We had witchgrass. Seeds were ripe. That's the seed rain. They are going to go into the soil. This is this box, represented by this box. Okay? Many of them are going to be dormant seeds. They're going to go into what we call the dormant seed bank portion. Okay? Some of them, as they come off the plants, will be predated upon. They'll be eaten. They'll be consumed. In some cases, they're going to be consumed while they're still on the plant. Okay? And so we use two terms that I'd like you to at least be aware of. Okay? Pre and post. Okay? Predispersal seed predation, okay, is one term that you'll hear of. Okay? And post-dispersal. So predispersal seed predation, when does that happen? If somebody says, oh, that species or that organism is involved in predispersal seed predation, what does that tell you about when seeds are being eaten, consumed, destroyed, you name it? Before they're shed, actually on the plant. Okay? And that's important. In biological control, it's basically, you know, trying to do the sandblasting before the seeds come out. Well, you're doing it on the plant. And in IPM, we're going to do a lab. For those of you in IPM, we're going to be looking at common burdock. But any of you, if you don't know what burdock, you'll know what it looks like. If you open up any of the seed heads around here, you're going to notice that the seeds inside a good number of the plants have been basically glued together by this moth, a lepidopteran moth, that basically lays its eggs on the developing flower in the summer. Remember, this is a biannual plant, so the first year those big leaves, rosettes. Second year it bolts, and what it'll do is the moth in June, July will lay its eggs in there. The eggs will hatch as the flower head is developing and the seeds are developing. The larvae then move in, okay, the different instars, and start feeding on the seeds, okay, of the burdock. And they do that by drilling holes through them, basically killing the seed right through the embryo, and also by gluing seeds together, forming hibernaculum, we call it, basically forming a little clump. And then basically throughout the winter, it's in there now, it's preparing to pass the winter in there and have enough food. And then come spring, it will pupate, and then you get the adults, and the whole cycle starts again. But that is this moth then is carrying out is a pre-dispersal. The seeds haven't dispersed yet. They're still in the seed heads. Okay? Seed predation. So that can happen. Okay? Here we're saying it's going to happen we don't have it. So predispersal seed predation would be happening up here. Okay? Seed rain is those seeds that are, you know, nothing's happened to them. They're fine. But now some of them are going to land on the soil surface and could be consumed. Who could consume things on the soil? Seeds on the soil surface. What would be some organisms? Sorry?
Small animals,
Birds can do it. What else? Mice, big. So small vertebrates. What else? Insects, beetles. And I've had students, I've worked with John Losey looking in BT systems, non-BT systems, looking at the levels of carabids, seed predation. It's pretty high. And that's why in some of the organic systems or integrated systems, we try to favor beneficial insects and carabid beetles. In some cases, for velvet leaf, for example, or for lamb scorters, some of them, basically removed 50% of seeds. We were working at a large-scale collaborator fields, and 50% of the seeds on the soil surface were being wiped out by carabid beetles. Okay, and so that was an issue. Does anybody know why? This is kind of a bit off the side here, but I just want to test your general agricultural knowledge. Why were we interested in knowing in a BT system? So remember Bacillus thuringiensis, okay? In BT system, transgenic corn, what the impact of BT was on carabid beetles. Because that's what the project, we weren't only looking at, we knew that these beetles feed on weed seeds. It's a big part of their diet. But why were we doing it in a BT system? Why didn't I do it in just a normal system? Why?
Because if that toxin killed those beetles,
okay.
they wouldn't be the
Good point. Now, is any BT, do you guys know that there are different BT for lepidopterans and so forth? We were working with corn rootworm. Does that ring a bell to anybody? Why that now makes sense? We weren't looking at borers. We were focused on corn rootworm, which is a big problem. Does anybody know what the corn rootworm is? As an adult, it's a beetle. It's a beetle. So the reason we were funded for this project, we were trying to figure out if there were going to be any indirect effects. So here you've got this toxin that's being constantly produced in the BT corn. Okay, the leaves disintegrate. Okay, and the worry was, hey, this BT toxin is out there all the time. Yeah, it does a great job on the corn rootworm, but we also have very closely related beetles that are beneficials that have nothing to do, with the BT. But could they also be impacted? And so we weren't only interested in diversity. Yes, these beetles get it, but also we were basically arguing that a lot of these beetles carry out beneficial. They're what we call beneficials in a system. They provide a very important ecosystem service. Okay, so sometimes you don't say, well, diversity dropped, that's not good. But also, okay, but did some really key players in that community drop out that were very important to sustaining that community? So the reason we were doing that work was exactly that, because the BT for corn rootworm was targeting beetles, and a lot of ground-dwelling beetles, carabids, are also tremendously beneficial. And we wanted to make sure that in a BT system that wasn't happening. What it turned out to be, we had BT corn. We had one where we were using Force 3G tefluthrin, which is an insecticide commonly used for corn rootworm. And we had a conventional system where we didn't apply anything. Of course, the corn was just devastated because, after second year of corn in this area, you're done. Okay? Turns out, just if you want to know, based on our results, that there wasn't a significant difference between the hybrid, the control, and the BT. However, where we used the insecticide, everything got wiped out because it's non-selective.
Is that because BT isn't that persistent?
That was one of the reasons. And this is concern. I mean, Monsanto, who had put out the project, was trying to say, hey, are we putting this out there? Could there be an impact on soil microbial communities? So if some of you know Janice Thies, Professor Thies, who teaches the soil ecology course here in CSS, she showed that microorganisms were not impacted. Because that was a concern. This thing's out there. Could it cause problems? Based on our work and others accumulating work, that does not seem to be the case. Certainly, carabid beetles were not, and part of the reason is that the toxin is broken up relatively quickly. Okay, so by far the more problematic treatment was using the insecticide. Force 3G wiped out. I mean, they were significant. What could have happened too is that the insects could have noticed that the beetles, they can smell, they can sense, and they got the hell out of the strips that we did spray. Because it was in different strips. So it could have also been a deterrent for them, saying, we're not going to be in there. I mean, this is going to kill. And they just went elsewhere. But everybody knows that if you do spray tefluthrin, it's not just going after the corn rootworm. It's just not selective. It'll knock off beneficial carabids. So in terms of rating it, I would rate, this is where I would say BT is actually has done quite a bit of an improvement in terms of, because some of those insecticides are pretty potent. And they affect the nervous system of insects and animals and so forth. So, if I don't want to, take sides. I know some of you are obviously, for or against, but just here's where we were trying to be objective. Let's see what the science says. At least from our results, that's what the conclusion was, that BT was not impacting non-targets.
So the weed population in the insecticide control was a lot worse than, or a lot higher than it was in BT?
We had the same control. There wasn't, we used the same standard control in the BT, because BT was just looking after the insects. The herbicide regime was the same in all three. And the weeds were, there was no difference in weeds, if that's the question. And again, we weren't focused on the weed component, but we were...
Like long-term, though.
Right, and maybe, right, long-term.
That's what I'm saying, because you didn't have the...
Oh, yeah. And that's where in organic systems, growers are very much interested in maintaining beneficials at field edges, which then, as you'll see, is going to be, in some ways, I'll argue that is good, but it's also bad, because that's where most of our weeds can also be if we don't take care of our field edges. That's how they move into our field. If you do a good job of keeping the grasses and, some beneficial vegetation, that's great. But if you're going to have ragweed sitting on the sides because you're not doing a good job, you're not going to say, oh, that's good because of beneficials coming in, because the problem of your weed issue is going to be way worse than any of the beetles taking out weed seeds. I mean, they don't take out everything. But they're, in an integrated system, they're important. That's what I'm trying to say. Okay? So, but that's what we were trying to do, was basically this. We were trying to lower seed levels in the fall by these carabid beetles. And this was one component of it. Okay? They can also, we do have some of the beetles can actually go, a few inches into the ground and go pull out seeds. Earthworms do a great job. And I think in this handout at the back, take a look at it it just came out August 25th. It's a news release from the Wheat Science Society, which is the main scientific body of all wheat scientists in North America. And just a news release on what earthworms do. They basically come out of their burrows, they grab seeds, and they're showing you grabbing a giant ragweed seeds. And they just pull them into their, burrows. Most of them they'll consume, but some of them they don't consume. And what you'll see is the next spring, 5, 6 seedlings coming out of these little holes. And you go, who planted that? It's almost like when you're planting cucumbers or something, you're putting a few more seeds just in case, and then you'll thin them out to leave. That's what that looks like. But it's some work some of my colleagues at Ohio State have done. But that is another way, a seed producer. Earthworms do a great job. They're night crawlers. They come out at night and pull these things in. And it's amazing how many they can pull. So take a read at that, and as well as an article on what you should be doing in your vegetable gardens in terms of the more you dig, the worse your weed problem is going to be. So that ties into the kind of some of the reality. But so what I want to say here is that most of your seeds are going to be dormant, and we'll talk about what does that mean. How do they become dormant? But just for now, just think of this. At some point, we're going to get a stimulus, some temperature fluctuation, light, and it's species dependent, that will stimulate some of this dormant seed bank, some of the seeds in there to germinate. And they move into what is referred to as the active seed bank. Okay? So they're primed to germinate. And they do. They get the right conditions, they germinate, and hopefully they're not killed at this stage, but they'll produce seedlings. Okay? You will get some of the seeds over time, and there's been a lot of interest in, can you put some numbers on decay and senescence? How many of these seeds die from decay or from pathogens or diseases? Okay? And this is really an important area now, particularly for organic growers. They're very interested in this because generally their soils tend to be more biologically active. There's a lot of microbial communities. There's a lot in there. And the idea is that perhaps there might be some pathogens that are not deleterious or problematic for your crop, but could go and are either specific for the wheat seeds and cause death. And that's something we're exploring now. And it can be fairly high, okay, in many wheat seeds. And one of my colleagues that just visited from Indiana was telling me that they've done studies, and they just published this, that if you just make a little cut on many, and I have to try this because this is kind of neat, on any wheat seed. So let's say, you just scrape it by, with machinery, or it smashes a rock, and then pathogens move in very quickly and can destroy those seeds very quickly. And he had high levels, particularly things like velvet leaf, which that would be fantastic. So there is an indication that one could, increase this component. So this is what part of the work in an integrated approach is. How do we draw the seed bank down? How do we get these dormant seeds to germinate so that we could do something to them? Either, control them with cultivation or put down a herbicide. Okay? We don't want them just sitting here. If they were all 100% dormant, cool. I wouldn't do a thing. But you're going to get 60% in here, 40% in here for many species. So how do we basically decrease the seed drain, increase predation, increase death and decay without affecting our crop? And how do we stimulate dormant seeds, a greater proportion of them to germinate so that we can control? That's the key. So keep this in mind, okay, in terms of a model that one can think about. Okay? What I want to really focus on is this, okay? They say developmental arrest, but when we talk about dormancy, and you may not have this because I just included it in, because I really wanted to show you that we're going to focus on this part here, okay? The dormancy onto germination. So we've got these seeds that are produced. What are the various ways that these seeds remain dormant? What is it about them? And not all dormancy is the same. That's the other problem. Or else we would be doing, okay? So let's talk about seed dormancy, because it is so crucial to seed banks and weed seeds, and it's very different from many of our other pests, okay? Because you're not going to get you can, but you don't get spores or an insect being in diapause for 40 years, in a rested state or in a quiescent state. You do. So there are a number of dormancy classifications. And we'll define dormancy in a sec. But basically, you can have what's called seasonal dormancy, opportunistic dormancy. That's not the dormancies I'm talking about. But I wanted to make clear that you understand what that is. Okay? Seasonal dormancy, okay, is based on seasonal rhythms of the environment. It's going to be a temperature, and again, they're related, but temperature and day length. Okay? And they're predictable year after year. So, for example, we're not going to be getting plants or weeds germinating in January in this part of the country. Not yet, anyway. Maybe down the road with climate change that might happen. Because it's seasonally, there's not the right time for them to germinate. Just the temperature, day length is very short. Okay, you're noticing day length short? You guys get up early, you see how dark it's getting? So we are approximately 10 days or so away from 12 hours, and then sunlight. And that'll be where? Just here in New York State? What's this 12-hour thing? Yeah, the whole world is going to be at 12-12, okay? Including the Arctic that have been at 24-hour sunlight, okay, are now, and they're dropping I've noticed they drop like 8-10 minutes a day. And the place I did some work on in northern Canada, they drop 4 minutes a day daylight. We're typically 1 or 2 minutes. Okay, just to put you in that mood, okay? And some of you are going to tell me I suffer from seasonal affective disorder, which is where you need to have some light therapy, which is actually a condition that people do suffer from. Didn't want to make light of it without a pun intended there. But seriously, it's one that I have seen. But this is plants. What I'm saying is you're detecting it, plants are detecting. So it's just not the right time of year to do this. Opportunistic dormancy is more of so this is very predictable. This is very unpredictable, where you go in, you till, in a sense, or you disturb the soil, there's a fire. And you notice some species are well adapted for fire. We got a plant called epilobium fireweed. It germinates right after fires. Jack pine, another example. Okay? And I'm sure you have some other, some species just, okay? Or in the desert. What happens? You hear about, in the desert, this after 10 years, we got a rainfall, and the place is in blooming. Last year it happened down in California. Okay? That is opportunistic. Okay? This is not the way I want to be thinking. There's specific environmental triggers that do this. So this is kind of an ecological classification. I mean, it still happens. It's important to be aware of it. But I want to get into more kind of the more closely related to the physiology. Okay? Physiological classification. Okay? A very famous plant ecologist named John Harper, when he was trying to think, how do you classify seeds? And, and this is the famous quote, some seeds are born dormant, some acquire dormancy, and some have dormancy thrust upon them. my friends, as John McCain would say, these are the three main divisions or classifications of dormancy in many wheat seeds. We've now developed this further, but these are it. So can somebody in your own language tell me, what is he talking about? What is he saying? That some seeds are born dormant, some acquire it, and some have dormancy thrust upon them. And in your own minds, and what does that mean? Can you give me an example of what they're meaning? So when I talk to most of you and say, oh, this seed is dormant, has dormancy, what comes into your mind as to, okay, it's not germinating, why? Why is it not germinating? What would be the kind of things you'd be thinking about why it is not germinating? Rob?
Well, it could be like the genetic makeup of the seed is like dormant at that time, so it's already naturally dormant. Like in the word dormancy, like first is where like one of those environmental factors or something happens in the seed bank where it says I have to be dormant.
Okay.
Or like we said, it's later in the season.
Okay.
Sensitive to cold temperatures or something like that, and there's no reason for it to die if I germinate now.
Okay. So actually, Rob's got the 2 out of the 3. Bang on. One is some seeds are born, as you can imagine. There's clearly some genetic component here that you could do whatever you want. You could grab these seeds, and a good example would be wild parsnip or ragweed seeds coming off the plant. You give them all the right environmental conditions, light, fluctuating temperatures. No way. If you don't wait about 8 weeks, generally speaking, 8, 9 weeks, you're going to get zero germination. So there's something genetically in there that obviously then translates to physiologically that doesn't allow them to germinate. The third one, dormancy thrust upon them. This is basically saying, hey, the seeds are viable. They would germinate. They're ready to germinate. They're mature. However, the environmental conditions outside or where they're found are just not appropriate for them to germinate, i.e., it's January. And, it's not 75 degrees outside, and they're not going to germinate. But as soon as you take those seeds and you give them the right environmental conditions, light, depending on the species, if they require light or if they need alternating temperatures, they will readily germinate. That's some of dormancy thrust upon them. Okay? So the first one is the terms we use, and you have them in your notes. Innate dormancy is some seeds are born dormant. It's innate, something within. Okay? The third one is enforced dormancy. It's basically the environment is forcing them not to go into dormancy because it's just not the appropriate conditions. They are mature. There's nothing to do with the fact that they're beyond this. Okay? Ragweed's has had innate dormancy. It's been 8, 10 weeks now in cool soils and so forth. But the environmental conditions are just not right. We have dark conditions or whatever. We just don't provide the temperatures. The only reason that ragweed wouldn't be germinating in that situation is because of an enforced dormancy. The tough one is the middle one. The term some acquired dormancy, what are they talking about? Can you give me an example where something will acquire dormancy?
Maybe talking about all the flooding, maybe that, too much water would the seed from germinating. It was just water.
Okay, so flooding situation. Yeah, Joe.
What if it was ready to germinate and then you plowed it under?
There you go. That's a good example. There are some wheat seeds that when they drop to the ground, okay, are primed. They don't have innate dormancy, and if the environmental conditions are right, it's the right temperatures. Let's say it's any of your cornfields. It's May. Good temperature, fluctuating temperatures. These seeds have sat in there. They're ready to go. There's no more innate dormancy. You come along and you're plowing. You take that top layer and you bring these guys down. Okay? There are a number, not all, but there are a good number of seeds. One of them, a good example would be something like, and it's not as common here, but does anybody know what corn spurgery is? Spurgula arvensis. It almost looks like a horsetail. It's a pretty neat plant, but that's a good example of one that has no light requirement. It doesn't really matter when it's coming off the plant, or it's near the soil surface. But as soon as you bury it, so it basically didn't have dormancy. As soon as you bury it, through cultivation or whatever means, it now becomes, you know, it needs light to germinate. Whereas before it could germinate in the dark, in the light. Now all of a sudden, being under there, it will not germinate. Okay? So, on the soil surface, here's the seed, okay, it was dropped and had stayed there, or it happened to remain there. No light would come. It would have germinated had you not come along. You know, if there was enough soil, say, just at least come. If you had come along, not turned it over, it now goes into the ground, and unless you bring it back up or give it light, it's dormant. Okay? And in some cases, you bring it up, and you provided light, and it's still, about half of them will still not germinate, where before 100% would almost germinate. You basically, you have induced dormancy. That's the...
So is the only difference between induced and enforced that enforced is via natural causes and induced is human caused?
Induced is typically some... Right, that's one way to think about it. But all that's limiting an enforced, okay, you know, in a seed from Germany is you give it the right environmental conditions. What you're saying here is that this seed was ready. I mean, it would have been, it didn't have enforced, it didn't have innate, it would have germinated, i.e. it was cleared. And it didn't matter if it had light or no light, it could have been just under the soil. And now by burying it, you basically have triggered a defense mechanism. This happens, for example, here's a good example, for ragweed. If ragweed doesn't germinate in the spring and it's still around in the summertime, after that first flush, it typically won't germinate again. It goes into secondary dormancy because of drought. And even if you provide water and so forth, you have to wait till next year. Okay? So some of these seeds that have gone into the soil would have readily germinated. Now you basically have induced dormancy in them. They said, oh, this is trouble. This is not good. I'm going to wait another year. So yeah, they're not going to germinate, but you've got them in the soil seed bank. Okay? So, innate, induced, enforced. Okay? Those are, and I'll give you some examples.
So
yes?
If dormancy is enforced, can the seed, as soon as the conditions change, the seed will be able to germinate?
Absolutely. Okay? Enforced is just the climatic conditions are just not there. Okay? And as Annie is saying, in induced, there's some external factor that's come in, either cultivation of some sort, okay, where you've now thrust this into dark conditions, and that's just not appropriate for those species. Don't get the idea that everything, as soon as you plow things under, oh, everything's going to induce. No, a lot of the species, you know, if they're innate, they're going to be innate for a while. They need a period, but often it's enforced. But there are a couple, and they just wanted folks to be aware that there's this category here, because these would not explain it, okay? We would look at these species and say, geez, they don't have innate dormancy. They don't have, you know, the right conditions are there. Why aren't they germinating? And then you would look at, well, what did we do? Was it a flooding event? That anaerobic conditions that they said, whoa, this is not good, shut down. You know, we're not going to germinate for you. It's drought, shut down. We're going to try it again next year, okay? Whereas if that flooding and so forth or cultivation had not occurred, they would have germinated under the right conditions. Now you might argue, well, the conditions aren't right. It's drought. There isn't enough water. Right. But for some species, they don't revert to dormancy. Okay? Velvet leaf won't revert to dormancy if there's drought. Even if the environment, it'll just sit there and wait for that. Whereas some of these other ones, there's a mechanism, and why they've been selected to revert back, that's a good question. They tend to be the real problematic ones. Remember, we want things to germinate as much as possible, not to get them to induce. We want to coax them out of these categories so that we could control them. Okay?
Tony, are you suggesting that the induced is some physiological lock?
It is. It does. In the induced, it's actually, that's what they're looking at it now from a chemical, physiological perspective. There is a change in chemistry. And it has to do with oxygen, CO2, you know, balance within the seeds.
So as you're saying, there's no imbibing it, there's no restarting.
No, you have to wait throughout the cycle. The ragweed that shuts down in the summer, if you take it and you give it all the right environmental conditions, it still will not germinate. Okay? So you basically have provided what we call secondary dormancy. Okay? You know? So these two are also referred to as secondary dormancy. The top is also referred to, if you see it in the literature, oh, this is primary dormancy. They mean this is innate. Wild parsnip seeds You take them off the plants right now, will not germinate. Because they're innate. They need about 6 to 12 weeks of after maturation period. That's just a physiology. The embryo is not mature enough in some cases. That's the other thing. Yes?
So when you're testing the germination rate of the, say you pull the soil core and you want to look at the seed bank, how do you replicate the certain germination requirements to get these seeds out of dormancy and do that accurately?
Okay.
Or do most of the summer annuals act the same way?
Prelim question. Because that's exactly what we face. It's good you thought of that. So, how do we do it? Do you get the question? Megan's saying, hey, if you've got all these, conditions in your seed bank, you've got seeds that are innate, you've got seeds that are induced, who knows what the mix is in there. And you're telling me you go out and you want to represent the sampling. I want to know what the seed bank is in my field. I go in with my, 6-inch core, bring it out. I'll, whatever. So what do I do? How would you deal with this?
You would extend that and just do it a number of years and hope that the average is going to be representative.
Okay. So do you know how do we actually measure seed bank numbers? You guess? Yeah, that's good, Joe, but you're not going to hold on to a job very, very quick. How is it done? Does anybody know? I mean, some of you may not know. I mean, this is not... yes, Steve?
Certain weeds have certain percentages of seeds that are one or the other.
Yeah, that's true. So you could kind of do that. But so if I were to set up an experiment, I'll say, okay, you're going to go in some of you have farms. Go back to the field Z there. Give me an idea of what the seed bank is. What would you do? Tell me kind of physically, what are you going to do? You're going to go and grab a sample, and then what?
Well, a simple way to do it is to take a soil sample and then give it all the right conditions, give it lots of nutrients and lots of light and water, and then count emergence.
Okay.
Well, if some of them have already been had induced dormancy on them, then
Yeah, you can't count it.
Right. So when you do that, that's a common way of doing it. Some of you as undergraduates or graduates, you probably have done that, will have done that, is you go and you grab, right, you got the soil, you spread it out on a flat, put it in a greenhouse or a grow chamber, provide alternating temperatures as best you can, okay? Anything that comes up, you note it, you remove the seedlings. So what that is doing is taking care of this. And what we also do is a lot of these tempered wheat seeds, like cold temperature, what we do is we do a first batch, let them go for a month or two, water. We then take the soil, let it dry out. We put it in a fridge. Let it sit in a fridge, simulating winter. It's called stratification. Okay? We let them sit for 2-3 weeks, a month, take them back out, start watering them, putting them in, coaxing some of these induced guys to come out, and allowing time for some of these innate guys. And so we could repeat this cycle of, good conditions, then cold, to basically try to get as much of this done. We will never get 100% of the seeds, because there will always be seeds induced, but it will be at least an indication. So it's not absolute. The only way to do it absolute, does anybody know what you can do?
Count seeds.
Okay, how? In the soil, and oh man, it's you can't do it. Don't get me wrong. Does anybody know maybe another way that we can do it? Tom?
Take your soil sample and then do a flotation to get all the seeds float out of the solution. And then you have all your seeds separated from the soil.
Right.
And you can divide them up to species. You can do germination.
You can actually do a physical separation, taking advantage of the density of seeds versus soil. Hopefully, you don't have a lot of organic matter, because that's it'll float to the surface. But that's definitely one way that it's done. And in our lab, Chuck Moeller's he's come up with a hydraulic. It's basically water taking. It's basically the same thing, where you basically provide water, and what happens is the seeds will float. The heavy seeds, some of them will velvet leaf, will be caught up first. There are various screens, and it's taking advantage of the soil. Mineral soil will just come down, and you basically scoop that up, and then you do the counting. But at least you don't have. You might have a few fragments and things in there, but you don't have just pieces of soil. And even there, you have to test recovery. You might lose some of the seeds, and so you have to kind of check what your error rate is. But that is tedious. It is long. And I don't know if any of the folks in here, usually we have our summer students that can tell you all about sitting there and doing all these samples. But so you could do it both by the germination tests, which is this whole growth chamber. That's the easiest. Most people, if you're just trying to get an idea, comparing one field to another, a system, that's probably not a bad way to do it. You know you're not going to get all the seeds, but it's a good indicator. But if you want, I need the numbers, I need as close as possible, then you probably have to do a physical separation, either through hydraulic or using compounds that allow the separation of the seeds. If any of you are interested in that, it's actually pretty neat methods there.
Just out of curiosity, why would you want to know how many thousands of seeds you have in square foot or an acre?
Oh, in some cases, it's not necessarily that you have, it's for example, we have a long-term, we'll go in 2 weeks, on the 23rd, 24th, we're going to go see a long-term organic cropping systems vegetable project that Chuck Moeller is leading. And he's got 4 different systems that you guys are going to look at that are, what a lot of growers would like to do. And what we're looking at is not necessarily the absolute numbers, but we want to know which system is not allowing seeds to come back. So relatively, which was the best system to cut down on the seed rate?
So it's for research.
It's for research, but we do have growers that, I mean, you're right. I mean, it's not going to be, but we want, I have growers that say, how did that system draw down the seed bank? I mean, they're still going to have numbers. Practically speaking, yeah, you're right. That I have 10,000 versus 5,000. But from a research perspective, yeah, in terms it's a quantitative measure for us to know which of these systems works. Or it doesn't allow wheat seeds to go in.
But that's also how you prove to a farmer that this management system
Absolutely.
is better than the other. That's the proof.
Listen, I have growers that I work with that even if velvet leaf is this size and may have 1 or 2 capsules, they're going to go in there and spray. A second spray, spot spray, and in some cases if it gets away from them. When I tell them that in general, geez, is it really worth it when you look at the balance? What they'll tell me, hey, I don't want any seeds going to the seed bank. And they know, and I don't blame them in that sense. So that's true. So if you can show them that, hey, this is being drawn down and this is another piece of a, little hammer. Think about all these management come together. There's no silver bullet. There's not going to be anything that I can tell you, oh, do this. That's panacea. But that is a way, and growers are relating to this. They know, hey, this is going to be hopefully money in my pocket down the road. But I know where you're coming from in terms of, what is that, is that really a big deal? But it can be, and we see big differences in the seed bank, okay? So we've spoken about this. You have this information. I want to get to, also referred to as quiescence. This is, generally referred to as dormancy. This is, most of you would have been thinking about what we call enforced dormancy. If you had known nothing about dormancy, I'd say, hey, why doesn't this seed germinate? Most of you would have said, yeah, because the conditions aren't right. And that is an important one. There's no question about it. We have a lot of wheat seeds that are not going to germinate in March just because the conditions aren't ideal for them. Okay? Having said that, I just want to leave you with this. There's 2 professors. They're a husband and wife team called Carol Baskin. Okay, the Baskins basically is I'm trying to think of Carol and Kyle. Do you know the other Baskin? John or they're very famous. They're at the University of Kentucky. They are the world foremost experts on dormancy of seeds. They have a book about this thick. If you want to know about dormancy, any plant, any tree, they're it. And I actually wanted to invite them to come to CSS to give a presentation, even to our Ag Sci students. So I'm going to try to see if I can get them in the spring. But they took this a step further and broke it up into these categories. I'm not going to ask, give me an example, but I just want to show you how, and I want you to tell me which of these categories, this is John Harper had them in this category, but the Baskins took it one more step, and they've got physiological, physical, combinational, morphological, morphophysiological. If anything, I want you to take out of this is, man, it's complicated. It's not straightforward. It's not easy. And that's why it's such a problem. Why I said if you can get these to germinate all at once, man, you got the Nobel Prize. It's absolutely true. Okay? Is that some seeds have what is referred to as physiological dormancy. Okay? So the key thing to keep in mind is the embryo in the seed actually mature? Is it developed? Or is it not developed? And what is making it, So what is creating this physiological dormancy? So what is it? The cause of dormancy, physiological inhibiting mechanism of germination in the embryo. Okay? The embryo is fully developed maturely, but it is dormant. There's something in there, whether it's chemicals inhibiting the germination, but it's not because the embryo needs more time to mature. This is what it's fully developed. It's ready to go. You remove whatever the dormancy mechanism is, and this will germinate, okay? And in your handout, I do have some examples of species that have physiological dormancy. Example, ragweed would be a good example, okay? Ragweed has chemicals on it that don't allow it. The embryo is developed. You would open it up, the embryo is fully developed, but it's just dormant. Physical dormancy. The only thing that's keeping seeds that have physical dormancy from germinating is a hard seed coat that's impermeable to water. The best example is velvet leaf. Velvet leaf, okay, the embryo is fully developed. It is non-dormant. It has nothing to do with the embryo. That's why if we scarify. To scarify means basically to nick a little bit the seed coat such that water can get in. There's no dormancy. And you know how we get 100% germination of velvet leaf? Like if you take velvet leaf and you put it in a Petri dish, alternate temperatures, and you give it all the right conditions, but don't nick the seed coat, you might get 30% germination. Okay? If you scarify, don't confuse it with stratify. Stratify means cold treatment to encourage germination. Scarify is basically physically, removing part of the seed coat without damaging the embryo, okay, to stimulate germination in some species. So the things like buckwheat, okay, field bindweed seeds, they're hard seed coat. Once you can let water in, the embryos develop, they're going to germinate, okay? So in our lab, we work a lot with velvet leaf. I need to get 100% germination. What do? I take the velvet leaf seeds, instead of waiting 2 weeks to have them, alternating temperatures, I take them, put them in a cheesecloth, 5 seconds in boiling water. Just do that. That's enough to crack open the seed coat without killing. That's why it's 5 to 10 seconds. You don't sit there 20 minutes. And you get 100% germination because the embryo is fully developed and it is non-dormant. Okay? These are probably the three most common. Then you have this combinational. Some species have both. They have a dormant, okay, embryo, but they also have an impermeable seed coat. So they're trying to put don't put all the eggs in one basket in a sense. And again, you have some example. Most of our wheat seeds are in this group here. Morphological dormancy, underdeveloped embryo. Okay? The only reason wild parsnip does not germinate, and we would say that was an innate dormancy, is that the embryo needs more time, even after it comes off the plant, to mature. It's just not mature enough. It's like a premature baby. It just needs time, additional time, okay, to really reach kind of its capacity. Think of it that way, okay? That's the only reason. So there's nothing wrong with the seed. It just needs that, and that's a mechanism. Why did some plants have that? That's selection. It's obviously worked because they're around, and we got a lot of them. And then you have this morphophysiological, which is really includes the underdeveloped embryo, and also, in some cases, some physiological, usually chemicals that inhibit, okay, germination in the embryo. So do you see why it's such a problem? Because we have, we're trying to figure out where each of these species fit in, our different species. So pigweed, right in here. Ragweed, right in here. And then we have things like velvet leaf, field bindweed. A lot of our morning glories are in the physical category. So that is the challenge. I wish, okay, we could say more. So it is time. I will end it here. Okay? Please read through the notes again.